This is a production of Cornell University.
Thank you very much, Maria. Thanks for the invitation and for the kind introduction. It sort of makes me feel aging. So yes, it's a pleasure to talk to people, and I'll talk to some more people after my seminar. I'm just sorry that I can't be there physically, because we were ruining the fact that over the last year we haven't been able to chat, and that's where a lot of science sort of chewing over and discussion goes on. But hopefully you can chew through what I'm going to tell you today, and I welcome any feedback, critical feedback or whatever. I've entitled my talk Studying NLR Immunity Networks Across Seed Plant Species, because we've been interested for quite a long time in how plants regulate their innate immune responses to host-adapted pathogens. And for the most part, we've been working in Arabidopsis for obvious reasons, as a genetic and also as a molecular and phenotypic model. But more recently, we've been asking the question as to whether the defense networks that we and others have defined in Arabidopsis, to what extent those defense properties hold for other unrelated plant species. So, for example, Solanaceous, and then even extending to the monocot plant species that have different immune receptor repertoires. And so I'm going to touch a little bit on that later point, but I'm going to tell you mostly about sort of work that we've done in the last couple of years to try and unpick NLR downstream signaling, so activation to signaling. So I want to share with you some of that, some of the results there. So just to set the stage, and this is really enshrined now in our models of innate immunity, both in plants but also in mammalian systems to some extent, is that you have two receptor barriers. The first is at the surface, the cell surface. These are called pattern recognition receptors, and they recognize various pathogen-associated or endogenous molecules, and that gives rise to a resistance response that we call pattern-triggered immunity, or PTI. There's loads of acronyms in immunity. I'm sorry for that. And then those pathogens that have adapted to a particular host genotype, they produce effector molecules that are delivered in various ways into the host cell. And those batteries of effectors enable the microbe to overcome this first defense layer, the PRR defense layer, and to gain traction, so to infect and cause disease. But those effectors then have driven the selection of this second barrier, the nucleotide binding leucine-rich repeat receptor immunity layer. And that's called effector-triggered immunity, because those NLRs are acutely sensitive to specific effector molecules, or what effector molecules do to endogenous host components. And that then triggers this effector-triggered immune response. And in recent years, there's been a lot learnt about how effectors trigger NLRs, and I think we're beginning to understand how NLRs then signal downstream. And I should mention that traditionally, these two immunity layers have been considered as rather separate entities, but there's some recent data, not from ourselves, but from other groups, if you're interested, which tends to suggest there is much more crosstalk between the PR and the NLR barriers than was previously thought. So one of the fascinating things about NLRs is that the same protein modules, or a similar sort of architectural logic, applies across animals and plants. And this is shown very nicely. For example, NLRs that you find in humans, and groups of NLRs that you find, for example, in Arabidopsis, but that applies also to other plant genomes. And the common principle is essentially that they have these central nucleotide binding oligomerization domains they have sensor domains that are often repeat domains, and you can see leucine-rich repeats occur quite frequently, that lend themselves for specific recognition and variation in recognition of triggering molecules. And then they all have these sensor domains at the end terminus. So there are various different domains, often involved in homotypic interactions. So those domains then come together to form a signaling module. And one of the fascinating things about plants is that, of course, unlike mammals, they have expanded the number and diversified their NLR repertoires much more greatly. And that's probably driven by this acute pathogen effector selection, because they rely on innate immunity for the resistance response. I should then highlight here, there are two main classes of sensor NLR in plants, so across plants. The one subclass has a toll-interleukin-1 receptor-like domain, so that's a domain that's also found in animal systems. And those are called TNLRs, or TNLs for short. And there are some examples here in Arabidopsis that will recur in my talk, because we've done some work on them. Then there's another group of sensor NLR that instead has a coil-coil domain, or a coil-coil-like domain. And there are some very good examples of those in Arabidopsis and other plants, for example, RPM1 and RPS2, well-studied CNLs. So those are two major subclasses of sensor NLR in terms of sensing effector changes. And I should also highlight, because this will come later in my talk, a subclass of NLRs that are not sensor NLRs, and they have a somewhat variant N-terminal domain that we call the CCR, or the helo domain, because it resembles helo domains that are found across organisms. It's a very interesting CC-like domain. And in plants, these divide again into two subgroups that are called ADR1s and NRG1s. And I will return to those, so sort of keep those in mind. And in plants and animals, these NLRs all work by the same principle. They're very tightly controlled molecular switches. So they, upon some sort of perturbation, it's an effector, or it's some host-modified molecule, or it can be a microbial pattern, that then triggers a conformational change, usually by starting at the leucine-rich repeat domain, a conformational change that involves usually nucleotide exchange, so ADP for ATP exchange. And then that gives rise to an activated molecule. And that then sets in train the downstream responses. And of course, because these are activating immune responses in both systems, so in mammals and in plants, they have to be very, very tightly controlled. And there are multiple checks essentially at the transcriptional and the post-transcriptional level to make sure that that most of these NLRs are kept in a controlled condition, so that it maintains homeostasis. But there are in both systems examples, very, very nice examples of where that homeostasis is lost and there's misregulation of NLRs. For example, here there are a number of SNPs found that underlie human autoimmune diseases, and these are due to a loosening of the leucine-rich repeats, which are normally tightly bound to the nucleotide binding oligomerization domain, and that then leads to a leaky autoimmunity, which can be very damaging. By the same criterion, there are a number of autoimmune plants that have been identified. Many of them are due to misregulation of the NLRs. And there's this famous one that was identified by my colleague Chin Lee in the UBC in Vancouver. And you can see the fitness penalty that's caused by a misregulation of this. In this case, it's a tear domain NLR, and that's due to a single amino acid exchange in the nucleotide binding domain. So there is it's important to keep these as very tightly controlled, regulated systems. So for a long time, we didn't understand what constituted a sort of active NLR, neither in the mammalian field nor in the plant field. And then there was a breakthrough in about 2017, and this is exemplified by this paper from the Russell-Vance lab, that showed the cryo-EM structure of a what's called an inflammasome oligomer complex that you can see here. And essentially that is seeded by one sensor NLR that recognizes a PAMP, and that then becomes activated and then causes an oligomerization of the helper NLR, which is NULP4. So NIP5 is the sensor, NULP4 is the helper, you could say, the ancillary NLR. And that creates this cartwheel structure that then acts as a scaffold for concentrating, for example, caspases and adapter proteins that then allow a concentrated enzymatic mechanism, which then helps to process pro-inflammatory molecules. So that's really quite well worked out now. And then later, so 2019, and then at the end of 2020, the cryo-EM structures of the two different types of sensor NLR, the coil sensor NLR, were resolved. And you can see this absolutely beautiful structure of an activated coil domain, so CNL, Rs1. This is an Arabidopsis CNL that recognizes a bacterial effector, actually indirectly. And that effector then modifies a protein that's bound to the NLR, and then that creates this oligomer structure. And then at the end of 2020, and we were involved with Gigi Chai, but I must give credit to Gigi Chai, whose team solved the cryo-EM structure of RPP1. This is a well-known Arabidopsis tier NLR. And at the same time, the group of Eva Nogales and Brian Staskovitz solved the structure of ROC1, which is a tobacco tier NLR. And I'm showing you the RPP1 activated complex. In this case, it's not a pentamer, it's a tetramer. But you can see the same principle in these NLRs, both the CC and the tier domain NLRs, is that what the oligomerization does is it assembles in an ordered way those N-terminal domains. So here it's the CC domains, and here it's an assemblage of the tier domains. And we discovered that this assemblage for the tier domains, of course, is a very structured one and very important for creation, as Maria said, of an enzymatic activity. So that was certainly an insight to the activation mechanism. I should mention that the structure also showed a domain in the C terminus of the tier NLRs that we didn't know from the sequence, so the primary sequence. So it only emerged with the structure, and then we did some HMM analysis and found that this C terminal domain that we call the CJID for C terminal jelly roll IG-like domain, that's a mouthful, and that seems to be present, so by HMN, present in a number, not all, but many TNLs. And what we found through a structure function analysis is that it helps solidify or sort of discriminate, I think, the specific interaction between the effector and the leucine-rich repeat. So this is the effector molecule, in this case ATR1, and this very, very C-terminal end of the tier NLR helps to solidify and sort of firm up that specific interaction. So that was something that was new that came out of that analysis. So if we go to a comparative between the Rs1 CNL activated oligomer and the TNLs, the work of Gigi Chai showed really quite compelling evidence that it's associated, the oligomer, the activated form, binds to the plasma membrane, where it can form a possible pore or channel. And so that's formed by these very N-terminal helices that poke into the membrane in Rs1. There was a very important breakthrough through two papers that came out in 2019 that showed that, by contrast, the tier domains of TNLs, as well as truncated forms of tier domain containing proteins, have an enzymatic activity, and that depends on an amino acid, so a key catalytic residue, and a similar environment that was found for a molecule that works in neuronal cell death and that was well characterized, that has this tier module. So there was this similar amino acid environment, and through amino acid exchanges, then this was discovered to be a catalytic active site for these tier domains in plants. And what was discovered is that a possible substrate is NAD, and then there were some possible candidate molecules that were products of this activity. And the activity required a homodimerization, homo or heterodimerization of the tier domains, and also, of course, the catalytic residue. And so this leads us to propose in the field that maybe these products that are generated by this tier activity are crucial signal intermediates to the downstream signaling. And if we go into then the structure again in more detail of the assembled tier domains in the tetrameric oligomer, so this is the activated form that has active NADase activity, you can see that there are two symmetrical homodimers that are associating through known homodimerization surfaces. But what's crucial is that the oligomerization causes a shift in the sort of a switch in those orientations, such that those two homodimers become asymmetrically arranged. And that's shown here. So this is the asymmetric, two asymmetric pairs. And it's that asymmetry that creates this active site and a hollow enzyme, so the NADase active site. So there's this oligomerization and this twist that creates an arrangement for a very efficient catalytic activity. So I think what the NLR is doing, or at least what these domains are doing, is creating a very highly kinetic, efficient, active enzyme surface that can work at low concentrations. And that was something that Gigi showed very nicely in the paper. So we've been studying for a number of years, together with Laurent Deland at INRA in Toulouse, an Arabidopsis tier NLR pair that's called Rs1 and Rps4, and it's really a well-known TNL pair that recognizes a number of different pathogens. And it's best known for its recognition of a bacterial, so a Pseudomonas syringae secreted effector called AVR-Rps4. And that's produced by this leaf-infecting pathogen. But it similarly recognizes POP2, which is a acetyltransferase that's produced by this root-infecting pathogen called Ralstonia. And we know that those effectors are intercepted by this NLR pair, this tier NLR pair, through a decoy domain that's at the C-terminus of Rs1. And that decoy domain resembles a Werke DNA binding site of Werke transcription factors. This is a large family of plant-specific transcription factors that orchestrate biotic and abiotic stress responses. And so that interception through the binding, or in this case, a POP2 transacetylation of the Werke domain, dislodges the heterodimer from its position at the DNA, and that's sufficient to activate the tier NLR response. And it's interesting that RPS4 has that CJID domain in its no, sorry, I beg your pardon. Forget what I say, it doesn't. RPS4 has in its tier domain the catalytic residue that shows that it's an active enzyme, whereas RS1 does not. So that fits with, perhaps in this activated heterodimer that, and it's thought that it stays as a heterodimer, there must be a reorientation of the tier domains of RPS4 such that it creates that hollow enzyme. And that's something that's really exciting to perhaps resolve for the NLR pair. So we've talked about activation of NLRs, in particular TNLs, and I've mentioned also the CNL activation mechanism, and the initial activation seems to be somewhat different. So what about the next steps in the signaling cascade? Well, we knew, and this was all identified by genetic analysis in Arabidopsis many years ago, that one crucial early component of TNL signaling, so that's downstream of the effector-activated TNLs as well as the auto-activated TNLs, is this lipase-like nucleus cytoplasmic protein called EDS1. And we knew from earlier studies that EDS1 associates with a number of TNLs in nuclei. So it seems to be at least closely associated with some TNLs. We never really discovered what the relevance of that association is, but it suggests that it's really proximal. And that would fit with the fact that it's required for all of the outputs that can be measured that are triggered by TNLs. And it's the founder member of a small class of lipase-like proteins, the two other members of which are PAD4 and SAG101. And these occur in seed plants, so they're not found in earlier land plant lineages. So they've evolved in seed plants to somehow act as these signaling components for TNL receptors. And we know that EDS1 forms mutually exclusive heterodimers with its two sequence-related partners, PAD4 and SAG101. And a lot of early work we did in relation to the function of EDS1 with PAD4. And we know that this heterodimer is important for conferring what's described rather sort of enigmatically as a basal immunity branch. And that's a branch that is distinctive and contributes to the TNL immune response. And I don't have time to go into it now, but I'm very happy to discuss what I think basal immunity is in this context. But just for the time being, this heterodimer is very important for the transcriptional defense reprogramming that's triggered by TNLs. And what it does is it prioritizes, in Arabidopsis at least, it prioritizes the salicylic acid defense pathway. And salicylic acid is a very important biotic stress defense molecule. And it does this by counteracting the activities of JA, and there's a bacterial mimic of JA, which is an effector molecule called coronatin. So it allows this boosting of the SA defenses and other defenses. So that's one very important output of this heterodimer, the EDS1 PAD4 heterodimer. So if we look at the structure of the two heterodimers, there's a crystal structure of EDS1 SAG101, and then there was a structural model made of EDS1 PAD4. You can see why these complexes are mutually exclusive, because EDS1 has a similar surface it forms, and this is dominated by a hydrophobic helix that fits into quite similar pockets of the two partners of SAG101 and PAD4. You can see that in more detail here. And that interaction, so from the analysis that we've done by structure-guided mutant analysis, shows that that's important for bringing the two molecules together and bringing together this very, God, it says my internet connection is unstable. I hope that doesn't crash. And bringing together what we now know is the action end of the molecule, which is these two, what we call EP domains. So these are alpha helical coil domains, and they need to be brought together to form a signaling active molecule in both cases. And I've put here catalytic pocket because these lipase domains are certainly in EDS1 PAD4 really absolutely classical alpha beta hydrolase domains, and they have a conserved catalytic residues. But when we knock those residues out, then we don't disable the immunity signaling at all. So our current thinking is that this is acting as a non-catalytic scaffold in the immune response. That, as I say, brings together these two molecules and the important C-terminal domains. And part of the evidence that showed that the C-terminal domains, the heterodimer, are really important is that they create a cavity, a very prominent cavity. And around that cavity are some conserved amino acids that we exchange, for example, in EDS1, and we've now done it in PAD4 and also actually SAG101. I have to say SAG101 surface is somewhat different, and that's probably quite relevant. And when we knock out, or when we exchange those residues, they don't interfere with the heterodimerization, but they interfere very strongly with the signaling capability of the heterodimer. And I emphasize that because there are some reports of EDS1 itself interacting with a number of transcription factors, and there was a recent report of NPR1 interacting with EDS1. That's super interesting, but I think it also has to be understood in the context of the fact that EDS1 without its partner doesn't do anything. So we can produce buckets of EDS1. If it doesn't have either one of its direct partners, PAD4 or SAG101, we don't get any signaling. So my feeling is that we really have to understand the heterodimer, and that's the active form, whereas EDS1 is more of the ground state, which could still, of course, be relevant. And there was one very interesting exchange that a postdoc, Deepak Bandarai, identified, which was an amino acid exchange of one of these conserved arginines in the EDS1 EP domain that borders this cavity. And that created, unusually, a weak allele, so weakly functional allele of EDS1. Most of them were sort of a full loss of function alleles. And it didn't interrupt the heterodimerization. It didn't interfere with the nucleus cytoplasmic localization of the heterodimer. But what it does is it doesn't efficiently transcriptionally reprogram the cells in response. So this is the response to PST-RPS4 that's recognized by Rs1, Rps4. This is a system that we look at the dynamics of the transcriptional reprogramming and the resistance. And so for here, you can see in this PCA plot, so these are differentially expressed genes at 8 hours, then at 24 hours, between this weak allele of EDS1 versus the null mutant or versus the wild type transformed line. And at 8 hours, the weak allele looks very like, in terms of differentially expressed genes, very like the EDS1 null. But by 24 hours, it's pretty much caught up with the wild type response. But that catch up is insufficient for it to confer resistance. And by gradually doing a sort of more fine-graded analysis of the critical time window that's required for this to efficiently, rapidly reprogramming transcription, it's between 4 and 8 hours for this particular immune response. So that tells us that there's this critical window in which that immune transcriptional response has to happen. And if it doesn't happen at that point, then it's essentially game over in terms of the pathogen overcoming resistance. And that's important because we base some of our later analysis on capturing what's going on in that time window. So I want to now tell you about a whole body of data, and I've just put it into one model here that was produced by ourselves and also many other groups. So Jonathan Jones's group, Chin Lee, there was a sort of a surge of papers, which was analyzing the TNL and, in fact, in some cases, the CNL reliance on these two heterodimers, EDS1 PAD4 and EDS1 SAG101. And as well as the requirement of TNLRs for these two different classes of helper NLR or RNL. So this is the helper NLRs that I mentioned before that fall into these two classes of the ADR1s and the NRG1s. And in Arabidopsis, there are 3 paralogs of ADRs and also 3 NRGs, except one Chinley's group has shown is probably a non-functional, it's a truncated form of NRG. And the upshot of all of that analysis that I can say, you know, relatively in a short way, is that different TNLs rely to different extents on these 2 downstream pathways. And we showed that those 2 pathways are genetically non-interchangeable. So you can't cross-use various components. These are really hardwired for EDS1 PAD4 to work with ADR1s and EDS1 SAG101 to work genetically with NRG1s. And in analyzing the Rs1 Rps4 response in terms of the different outputs, so the resistance to pathogen, as well as a ability to trigger host cell death, which is an important component of the ETI response, we found that the EDS1 PAD4, and I've explained before, this is really transcriptionally very active. And it's very, very important for the resistance. And many TNLs use this preferentially for the resistance response. And that tends to mask this other pathway, EDS1 SAG101, which at least has a more efficient capacity to trigger the host cell death. So it looks as if qualitatively, these are not identical outputs. And it would be very interesting at some point to discuss about, you know, what is the decision-making between this branch or this branch. So that's all in Arabidopsis. So the Arabidopsis TNLs can go this way, or they can use a bit of the 2 branches. In tobacco, so N-benthamiana, where we can reconstitute resistance responses. So for example, the TNL response conferred by ROC1 that recognizes a bacterial effector ZOP-Q. And this has been very well sort of worked out as a system to look at TNL responses in N-benthamiana, as well as if you express, say, an Arabidopsis TNL, that will also work in the N-benthamiana system. That relies entirely on this branch, the EDS1 SAG101 NRG1 branch. And this, I think that's interesting in that it doesn't utilize in the TNL immune response this branch, ADR1, EDS1 PAD4, even though there are conserved and functional orthologs of these components in the tobacco system. So bear that in mind for what I'm going to say later. So Dimitri, so we have this genetic evidence of these 2 branches or nodes that are operating downstream of TNL activation. And Dimitri Lapin, a really talented postdoc in the lab, together with Vera Kovakova at the University of Cologne, did a phylogenetic analysis of the EDS1 family across seed plants. So they identified orthologs in about 45 different species, spanning the angiosperms and the gymnosperms, where we knew that these genes occurred, and then analyzed the different lineages. And we found that whereas EDS1 and PAD4 and the ADR1s occur across the angiosperms and the gymnosperms, so they're really widely distributed, SAG101s and the NRG1s have a limited occurrence, and they've been lost selectively from a number of angiosperm, a number of dicot lineages and from monocot lineages. And this fitted with the occurrence of TNL receptors. And that was shown really very elegantly by Blake Meyer's group and then later by a number of other groups. So the occurrence of TNLs is limited. And this strong co-occurrence signature between TNLs, the SAG101s and NRG1s suggested to us that there might be a co-function, so a Coevol function in this particular node. And that was also suggested that perhaps these, you know, because they're Coevol, they work molecularly together. And I just want to show you one piece of data from a paper that's already published, so you can go into it if you're interested. But I think it illustrates how these modules have to work. And that is that it's only components from the same clade within this module that seem to work together. If you use cross-clade combinations, they don't operate. So here, this is the cell death response measured by ion leakage, quantitative cell death, in the tobacco system, in which we've reconstituted a tobacco plant in which we've CRISPR'd all of the EDS1 family. So this is called MBEPSS. So we've got rid of all of the endogenous EDS1 components. And then we've co-express various Arabidopsis components to see whether we can reconstitute the TNL immune response. And this is ROC1 recognizing ZOCQ. So you can see in the wild type N-benthamiana, if you deliver ZOCQ, you get this very nice cell death response. So that's our sort of positive control. And that's clearly ZOCQ dependent. Then if we start adding in various components, so here we've got solanaceous EDS1 with a SAG101 form that we knew was functional. That also creates a cell death response, so a ZOCQ dependent cell death response. And then we started adding in various components, so EDS1 with PAD4, SAG101, and it was only when we added EDS1, Arabidopsis EDS1 with Arabidopsis SAG101 and Arabidopsis NRG1, it could be either of the NRG1 functional isoforms, that we get that reconstituted cell death. And that was very striking because it suggested to us that really you need that molecular compatibility that's not provided between clades for that signaling system to operate. And in this system, the PAD4, whether it was expressed with EDS1 together with ADR1s, didn't work. So it wasn't that we were using the wrong combination of that. So that emphasizes that in tobacco, you're really feeding TNL resistance through this SAG101 NRG1 clade branch. So again, I just show it here that this I think it provides some good evidence that we have this co-functions of the TNLs with EDS1, SAG101, NRG1s. And that represents a resistant cell death signaling module. And I just wanted to reiterate that I think it's super interesting that the CNLs don't seem to require these components for resistance and cell death. And it's thought that maybe the CNLs, through this pore forming activity that I showed for ZAR1, maybe that's a common way in which they can trigger the immune response and cell death. Whereas the TNLs require these additional components to fulfill the immune signaling. So they're working in rather different ways to achieve the same response. And so, okay, oh gosh, sorry, I'm going a bit over time. I'm gonna, so we did some analyses where we did mass spec analysis where we used various components, whether it's EDS1, SAG101 or PAD4 in Arabidopsis, and asked what we pull down at an early time point. So this 4 to 8 hour time frame where we knew at least there should be some sort of activity that is important. And we found initially that the NRGs, and this is in nuclear extracts, interesting, NRGs interact with EDS1, so this was the bait in this case, and this is a quantitative proteomic peptide analysis. So they were coming down with EDS1, SAG101 and PAD4 also, obviously highly enriched. And then in reciprocal analyses, we used, instead of EDS1, these are all native express tagged versions complementing the mutants. We use a comparison of SAG101 YFP tagged with PAD4 YFP tagged, both in a background where the other branch would not interfere with the complementing protein. And these showed, and this was done at 6 hours, so it's sort of going a little bit more to an earlier time point. And in this case, we were triggering with PFO, which delivers AVR-RPS4 very cleanly. And the mass spec analysis showed that with SAG101, we enriched the NRGs, as well as, of course, EDS1. Whereas with PAD4, we enriched more the ADRs that did not come down with the SAG101. So it looks as if this genetic discrimination of these pathways is borne out also by some sort of molecular discrimination. So there seem to be these induced associated complexes of these EDS1 family heterodimers with different helper NLRs. And then a crucial experiment that Joanne Feehan did, and this is in collaboration with Jonathan Jones' lab, so Joanne's in Jonathan's lab. And this is one where she used a complementing, so this is in a different background, a complementing NRG1 tagged transgenic line. And she used that then to IP extracts from extracts that are being triggered by PFO alone. So this would trigger a PTI response, essentially, or PFO expressing AVRPS4 that would trigger a TNL ETI response. And what she found in that IP, so when she IP'd NRG1 with FLAG, is that it's only in the ETI triggered response that she is able to pull down EDS1. And then we looked at the mass spec data here, and that was associated also with SAG101, not with PAD4 or with ADR1s. So this says to us that there, and this is at a time frame before really, it's just at the cusp of the transcriptional reprogramming. So this says to us that an ETI signal, a trigger, is required for this some sort of induced association between EDS1 and SAG101 and NRG1. And I think that's suggestive that the PTI alone is not sufficient. And I'm going to just go cut now to just a final slide in which we've used the tobacco system, so a slight tweak on the tobacco reconstitution system that I showed you before, where we could follow the timing of the TNL activated signaling a bit more precisely. And we found there, so this is in the ZOC-Q triggered system, so this is the ROC1 resistance response, that even though we co-express earlier all of these components, you need, again, the TNL trigger to observe an association between NRG1 and EDS1 SAG101, and that comes in this case at about 12 hours. And that does not occur when we use PFO alone. So both in the Arabidopsis system and in the tobacco system, it looks as if we have this ETI induced association. I think that's important for the resistance response. And then we made a number of mutants in the EDS1 heterodimer, as well as in a modeled form of NRG1 that was modeled on the oligomer of ZAR1. And all of these mutations that we made knock out the signaling, but only a portion of them knock out the interaction between EDS1, SAG101, and NRG1. And those are this P-loop defect. So this doesn't allow the NRG1 to, we think, probably oligomerize, activate, but not these N-terminal residues that would resemble the residues that are important for something that could resemble a pore of ZAR1. So those do not interfere with the interaction between NRG1 and EDS1, SAG101. But the EP domain residues that are crucial for signaling also interfere with that. And so from that, we come to some sort of scheme or model where we have this activated TNL. It's producing an NADA's activity. We think it's giving rise to some signal intermediate that could bind either to EDS1 SAG101 or to NRG1 or both. We need the activated form of NRG1 as well as the competent EP domain of EDS1 SAG101 for there to be a ETI-induced association. And we would argue that that association is likely to be an important step for the activation of the defense response. I think we're gradually sort of getting more sort of unpicking the requirements for the response. So I'll stop now because I know I'm over time, and I won't talk about this. So I'd like to thank members of my group, fantastic group of people, particularly Dimitri, Tinghua, who contributed to the work that I'm talking about, also Yoram and Patrick, and my wonderful, wonderful technician, Jacqueline, and all of our collaborators, Carsten Niefen at the University of Cologne, Laurent, I've mentioned the work here, Gigi Chai in Cologne, also Haitao Ku, and I mentioned some analysis that we've done with Joanna and Jonathan. So thanks very much, and I'm very happy to take some questions if we have time.
Thank you, Jane. It was very good. Thanks. This
Thanks.
is from Saeed. Thanks for a great talk. What factor do you think is important in choosing between the two paths? The type of pathogen or the effector or something else?
That's a great question. We don't know. Well, you always say it's a great question when you don't know. We don't know the answer. I have an idea, and I think that it might be the strength of the ETI signal. So if, for example, the TNL is, so in those tissue, in those cells in which the TNL is being directly activated, I think that then that should signal through the EDS1, SAGR1, NRG1 branch. Whereas we know, and this goes down to this basal immunity. So I see the EDS1, PAD4, ADR1 branch as the basal immunity branch. And I suspect, and if you look at the data, and we talked about this in an annual review if you're interested, at least an argument, so various pieces of data which suggest that the basal immunity branch is acting as a reinforcer in cells around that immediate effector-triggered focus. So I think they're probably working to different extents. So if you have a weaker TNL response, you might then tune it into the EDS1 PAD4 basal response. But if you have a strong one, then you go into the other branch. So that's my thinking, but you know, we might well be wrong.
And I think Gillian has her hand up for one.
I do. Thank you.
Hi, Gillian.
I'm always a big fan of choosing a model and discovering everything one can about pathways and so on. Unfair question. How do we translate this to the field and control disease in the field?
Yeah. No, it's actually, I'm going to be unfair as well. I think the most power we have in terms of engineering resistance and translating it to the field, I don't think it's necessarily in the signaling components, but I think it's in the NLRs themselves. And there's just some super work going on about in terms of designing integrated domains in such a way that you can broaden the recognition space without auto-triggering, you know, upsetting the balance of the defense homeostasis. So my feeling is that if you're doing translational science, and now's a time, if ever there were, the opportunity, the understanding the NLRs and the different types of NLRs and how you have complementary sets of NLRs is the best way to go for engineering robust resistance. One thing I think was very surprising, at least to me from our analysis, was that we didn't expect there to be such selectivity in the signaling components downstream. So that was a surprise to me. So if you wanted to mobilize that cassette into another unrelated plant, you'd have to take all of the co-evolved components with you, and that's what it tells us. And we're doing some analysis in rice, where we're CRISPRing a number of these components, and that's to really be able to understand, well, what are the basic components, because rice doesn't have TNLs. So that's a system in which we can, I think, unpick how these circuits work in a crop plant. So that doesn't answer your question, but it gives my preference as to where I think the field should be going for resistance.
Thank you so much.
There's a question in the chat from Jen, which is the EDS1 SAG101 NGR complex in the nucleus?
That's another really good question. I think there's an active pool in the nucleus, and that's from a number of different studies that we've done, and we're doing some localization studies, and we think that yes, there's an interaction in the nucleus. That doesn't mean it's the only pool. And in fact, you know, Arabidopsis SAG101 is nuclear, tobacco is nuclear cytoplasmic, so it's not an absolute. And also, Jeff Dangle's group has just published a bioarchive paper which claims that NRG1 can autonomously form a pore, a calcium channel, in fact. And that could be the case. And I didn't have time to go into and that would be at the plasma membrane. But they don't consider the fact that you need, I mean, at some stage, you need this association or co-function with EDS1 SAG101 for the NRG1 to do its job. So I think that's something that really needs to be understood, and whether that's nuclear, and maybe that then modifies NRG1 in such a way that then afterwards it can go to the plasma area. That's a perfectly possible scenario. We don't know.
One from Adam. Are there examples you know of hybrid incompatibility arising from mismatched signaling components, or is it all NLRs? If no,
that's a great question, Adam. There are some. So the best one that comes to mind, so most of the immune-related hybrid incompatibility, so this is genetic mismatches when you cross various accessions of Arabidopsis or even interspecific crosses between plants, you get this autoimmune response. And many of the causal loci are NLRs, but Detlef Weigel, for example, identified RPW8, which is a helodomain membrane-associated protein that is somehow involved in basal immunity signaling. The components that we're interested in, we've never identified those as being causal for the HI. And there are other components that have been found, certain metabolic dysfunctions that then are somehow leading to an autoimmune incompatibility.
One from Lei Yun to everybody. Thanks for a wonderful talk. What is the biological relevance of EDS1 binding to TNLs? Does EDS1 affect the organization of the resistosome, or nuclear EDS1 TNLs might have a transcriptional regulatory activity?
Yeah, great question. Yeah, we think that there's probably a transcriptional activity. We identified EDS1 interacting and EDS1 PAD4 interacting with a couple of TNLs in IP experiments. There was a super experiment, a turbo ID experiment done by Dinesh Kumar, then UC Davis, fairly recently, where he used tobacco N, which is a TNL protein, and he pulled out, so turbo ID is where you get this biotinylation of associated proteins, and it's sort of in vivo, so it's before you break up the cells. And there he identified all sorts of interesting things. EDS1 was among them. So that reassured us that, yes, it's probably there. And then to your second part, is it part of this resistosome? I think, you know, if you look at the mammalian literature, you think, yeah, there's got to be this concentration. So actually that's something that we and probably a whole load of other people try to identify, these higher order complexes. That may be some higher order signaling scaffold, because, you know, you're bringing together, so the principle is you're bringing together these components to concentrate them to then act as a sort of a kinetic downstream process. So that it wouldn't surprise me if that's also the case in plants.
I think that's all, which is perfect timing.
Thank you very much. Thanks for hanging in there.
It's the time to move to the next session. So thank you very much, Jane. It was a great seminar. We really appreciate your willingness to visit us virtually. And we'll continue with the schedule, which is the students and postdocs at 1:30. But please let Jane have a small break. Remember, she's just being beamed around from Zoom room to Zoom room, and she might like to get a drink or a quick break. So
thanks very much. I'll join you. I've got a new code, haven't I? So I'll join you
in the new code room. Yes.
Okay, cool.
Thanks. And thank you for a great talk.
This has been a production of Cornell University, on the web at cornell.edu.